Hi, everyone. We're just waiting for Richard Novak to log on and then we will get started. Um, Anna will maybe Anna will maybe sign on in a little bit. She is at an appointment, so we will see, but shortly we should get started. We'll see when Richard comes on. Maybe him. Let me see.
La la la.
Everyone.
Richard, how are you?
Good.
I think we can give it maybe just a couple more minutes and then we can hop on. Richard, I'm Christy. I work in Curtis Coughlin's lab. I don't think we've met, but you probably
No,
know Curtis.
I'm Dr. Curtis Coughlin. Nice to meet you.
great to meet you.
Anna will try and jump on in a little later, but I figured we could just kind of get started. I know you were in talks with Cure PDE, and I know everybody's interested in what your company does. So I was just going to hand it over to you and kind of let you take the lead. I don't know if you prefer, if you want to do like a Q&A at the end, or if you want people to interrupt you as you go. How would you like to structure it?
Yeah, I mean, I think just talking with Anna, I think the idea is just to do a kind of like a 15-minute overview and then basically spend the rest of the time answering questions, diving into any details that people might want to do. So
Perfect.
if that works. Yeah, also happy to be interrupted. I really don't care. But I think that's just what we discussed. If that works for everybody, because I think there'll be some questions that get answered sort of along the way. I think that was the idea.
Okay. Sounds great. So I'll hand it over to you whenever you want to get started. Feel free.
Perfect. Let me share my screen here. Let me know if that's showing up. Great. Okay. Get rid of this. All right. Perfect. So anyway, thanks for the opportunity to speak with everybody. I'm Richard Novak. I'm a co-founder and CEO of Unravel Biosciences. Today we'll be talking about the project that we're starting with the Cure PDE Foundation. And really, I was asked to present on what we do in general, high level, and then kind of dive into what this current study is doing, and sort of the potential impacts for the PDE community. Again, happy to answer any questions. We're gonna have plenty of time for that. So definitely, yeah, I'm here to answer any and all questions. So what we do, and I'll talk about a little bit more kind of how and why in the coming slides, but we're a biotech that spun out of Harvard about 3 years ago, and we're essentially bringing this engineering approach to drug discovery. And we started a program around 2 years ago, a year and a half ago, called Rare Shift. And the idea is really rapid repurposing for rare diseases. So kind of innovative, highly effective therapeutics, but basically using existing drugs to do that. And so then this is really what we're starting off with Cure PDE. So the study has already started. This is not theoretical, but we're just in the beginning of recruiting participants. So definitely, I know there will be some questions around that. So the premise of Rare Shift and Unravel in general is the following, which you all know: drug development really doesn't work well for rare disease patients. It was really the current workflows by big pharma have been developed around these large blockbuster diseases. The challenge is it's not that rare diseases are rare; they're very fragmented. So there's a lot of patients just in the U.S. It's basically 1 in 10 people have some sort of rare disease, which means that everybody's touched by it in some way, either directly or very closely. The issue is it's not that it's few patients, it's a very diverse, very fragmented group of patients. And then when a typical drug costs, upward of 200 million, kind of bare minimum, to actually develop the drug, not to even pay for failures, it becomes completely impossible by and large for most diseases. And so this is drug development as it's done now, target, some sort of preclinical development shown by a mouse. Then you get to the patients and you see, does it actually work? And then you have a drug in the positive case that it does happen. This really stinks. It takes many years. Most of these approaches fail, and you can't really take 10 shots on goal with a handful of patients. It just doesn't make any sense. And so the reason behind this is really the premise of Unravel is people are trying to treat disease, and diseases are not patients. And so these are all examples of really great schematics of various diseases that we work with, or tangentially or directly. These are sort of the ideal cases. These are all descriptions of what a disease is, but not any one patient. Patients are not diseases. Individual patients respond to treatments very differently. Every clinical trial has non-responders. Sometimes they have responders to a drug. And this has been known for a couple centuries now, that you treat the patient, not just the disease. And that's actually a picture from our lab. We have pictures of some of the families and children that we work with. Mostly we work on neurodevelopmental disorders, so it tends to be children. And so what Unravel has done is really revolutionizing how we go about this. And you can see that the order of these pieces is quite different. We start with patients, and we work iteratively within patients as much as possible, going to mice, we use tadpoles as well, other preclinical models, find targets potentially, and then develop the drugs. That's the end goal in all the cases. But you can see that we put patients really first in this whole discovery process. And essentially what we have, and I'll talk about our platform, half of what we do is computational and half of it is experimental and clinical. But really, if you were to summarize it in lay terms, we forecast how a patient responds to treatments, patient by patient, because it's individual patients that respond to treatments. So to do that, we have this patient RNA signature, and again, we'll talk about how and why and all those good things. And then similarly, a drug signature. What does a drug actually do? Not what it was developed for, but what is it really doing? And then using computational magic, we ask the following questions. Will it help this patient? So that's more of a personalized study. What we're doing in this study is, can we treat all the patients within a particular diagnosis? It may be multiple drugs are needed. It may be just one. We don't know, right? But we can start asking those questions today, and not in a clinical trial where something fails. Are there better treatments than current standard of care for PDE? Obviously, diet, pyridoxine, supplementation are kind of starting points. There's clearly a need for additional treatments. Are there different targets, different mechanisms? And again, we can discover those sort of in parallel. And then it will, a given intervention, whatever it is, doesn't have to be the one we're trying to develop, but will actually hurt a patient. So the toxicities, the adverse negative responses that you do get sometimes, unfortunately, right? So can we predict those? So can you predict the good and the bad and sort of the better ways of going about it? And so at a high level, this is sort of our mission, vision, values statement. We do this because patients can't wait. We're essentially saying we can't wait for 10, 20 or more years to understand a disease, because some of those patients may not have that time. I mean, we work with some really, really sick patients oftentimes. And that basic research is critical, but it doesn't directly necessarily tell you how to actually treat the group of patients. And so we're changing drug development by basically, and this is something that we did as a biotech, having this platform for clinically de-risking new targets, new mechanisms for treating patients using existing molecules, right? So we're doing this just for our own benefit as a for-profit biotech. Can we make drugs that actually work? Because if yes, we will proceed faster with lower risk and make more money, right? So it's like from just a strict capitalist perspective, it makes sense to make drugs that work, but then patients benefit in the process. Now, what's really unique about our approach is that this clinically de-risking of new targets, new mechanisms using existing drugs, that's repurposing, right? Repurposing is really taking that more to a commercial logical conclusion. That may not be the case for a lot of molecules, but Could it help a patient today just through standard of care changes, off-label use, even clinical trials without the intent of actually getting it approved? Yeah, you could do that, right? And so that's actually how we do it. Take an engineering approach to drug discovery. Try it and see. And then we work with patients no matter how rare or unique. I mean, we work with some patients that unfortunately have, three ultra rare diseases in one. And is it three ultra rare diseases? No, they're interacting in some unique way that's unique to that, one in a trillion patient. But yet for us, it still makes sense to work with these true N of ones for on the discovery side and repurposing side. And then we share everything with everyone. We just preserve the right to commercially develop whatever may be attractive commercially, but we believe in advancing the science, advancing the understanding of these disorders, and sharing everything back with the foundations or individual families if it's like a personalized study. And to that end, about a couple of years ago, we started this new business model. So I mentioned RareShift is really rapid repurposing, and it's anywhere in the world. And that's really structured as a just cost reimbursement for us. Like our price is to you is our cost, right? And that's what we're doing with the foundation. In return, we preserve the right to commercially develop potential discoveries, whether it's targets or molecules, through our biopharma arm. But this essentially ties that scientific discovery to clinical benefit. And then we're essentially a rare and ultra rare disease biotech partner as well. And so what we did over the years was actually work that came out of Harvard. So my co-founder and I were at the Wyss Institute as staff, managing a lot of these DARPA and other Department of Defense programs, really aimed at very rapid treatments, rapid discoveries for completely crazy diseases and scenarios. And so we developed all these technologies that we've now bundled into Unravel. So BioNav is really that computational piece. And it's a model of human health. Think of it as Google Maps for health. You use AI to understand using RNA networks where a patient is. That's your starting point in Google Maps. Your destination is a healthy control or a population of healthy matched controls. And then we simulate all these different paths. So it's hundreds of millions of different paths and networks. This is where AI is actually quite effective. And we do the same thing not only for this model of health in the patients, but also we can screen 40,000 molecules. These are all existing molecules, about 8 to 10,000 you could actually give to a patient, whether it's a prescription drug, supplement, nutraceutical, it could be food, pyridoxine is in there, things like that we can see. Then what we do is we do go straight into animals. We believe that for a lot of the symptoms that patients care about, that we care about as drug developers, we're asking questions around seizure, neuromotor function, cognitive function, sleep. Sometimes all of those functions, you really don't have good models in a dish, but you have that in an animal model. And so we go there as needed. We work with Jackson Labs on mouse models through their rare disease translation center. And then what we do, we have a chief medical officer, and we're actually about to start 2 clinical studies. We develop clinical proof of concept studies. Actually, one foundation is funding their specific, it's a 5-patient study, very small. Just does it actually work in actual patients, right? That's why we're doing this. How can we test if it actually works in a patient? Well, you have to test in a patient. And then after all of that, that's only when we develop new molecules that could work even better, that are now validated for that patient population. And so what we're doing, and this is again going more deeper into the study that we're starting right now, we use nasal swab. So unfortunately, everybody knows how to swab for COVID. It's literally a COVID test kit. We send these kits out basically anywhere in the world. A couple countries we can't ship to, obviously, but by and large, most of the world. And then we essentially use the cells that are captured from the patient and from the match control to get genetic insights into the condition. Not based on DNA, which is probably how all the patients in this community were diagnosed, but based on RNA. I'll talk about that in a second. And then we run it through BioNav, through that network model of human health, that Google Maps for health, and it tells us what's going on in your disease. Are there new ways of treating it? Are there unusual circumstances for patient 1 versus patient 2? Predicts response to all those 40,000 molecules, and it potentially can give you treatment options. Again, we cannot treat patients, but that advances the understanding of the disease and how we could be treating it. And so I mentioned RNA, And so the idea is that RNA, unlike DNA, actually tells you what's going on in a disease. It captures diet, medications that patients are on or not on, even things like microbiome in your gut, sleep, activity. All of these things help us understand disease and all the dynamic changes that happen in a given patient, good or bad, right? Whatever it is, all the other genes in your body have some role to play. And what's really exciting is that we had this innovative observation that nasal swabs, again, these are COVID test kits, right? So here's one of those swabs. They capture the brain and body state. It captures most of the RNA that's coding and non-coding, and it actually covers neuronal genes in the nose, which is interesting. So we see all the like strictly neuronal and synaptic genes represented. And then using our network analysis, we can even extrapolate from that. Because there's a preservation solution, we can ship at room temperature and it inactivates pathogens. We've been able to ship all around the world, every continent except for Antarctica. No penguins yet. Is actually out of date. We have increasing these numbers, hundreds of samples a week. And we've shown that this approach, this is actually not, this is just for the platform using other data that was given to us. We showed that we can actually predict clinical outcomes in this is for COVID and is looking at death from COVID, but in response to very specific, closely related molecules. And turns out there was a very significant difference between certain molecules in the statin class. These are lipid-lowering drugs. Turns out some are very effective against COVID, some are not. Some actually increase the risk from COVID. And we published this a couple of years back. We had a pretty good predictive outcome of this. And then very recently, we actually worked with the SYNGAP Foundation, and this is actually very similar to the study that we're doing with Cure PDE right now, sampling a whole bunch of patients. So in this case, it was 20 patients, all with a SYNGAP1 diagnosis, but all these different variants, different exons in the gene, different ages. This is the patient population, right? It's kind of a mess, and that's the reality of doing clinical research. Well, what's really exciting, we can look at how do individual patients respond to all these different drugs, right? And we can start clustering them, and we can start seeing here's cluster 1, if you can see my cursor, kind of that top left. There's a large middle cluster, cluster 2, and then there are kind of these, I would say, oddball ones, cluster 3. They actually do form a cluster for more technical reasons. They're not part of cluster 2, but you can start seeing that even despite all these differences, there's no pattern here, by the way. We searched pretty hard. Despite all of that, despite identical genotypes, people can have very different drug responses. And by the way, this is actually pretty accurate. So this is the kind of work we're going to be doing with your community, looking at drug responses based on all the medications, everything that a given patient is taking, all their background genetics, all of that makes them who they are, right, as patients. And then their treatments may differ as a result. And obviously for each of these subgroups, we can do a deeper dive, figure out the best clinical strategy for clinical translation, whether it's repurposed drugs or new ones. And then on the personalized level, this works. So we have already shown clinical impact. In here are 2 cases that I can talk about publicly here, but one it's a patient on the left. You can see this is a boy with Rett syndrome. So this is actually before we started working with them. We did a podcast series, which by the way we do pretty broadly with a whole bunch of groups. But we identified Vernistat, which is an oncology drug that has been used in again for certain blood cancers, and showed that actually could work really well in animal models. And we were developing it for girls with Rett syndrome, and it turns out it actually could be quite effective for boys who are quite different and much more severe. I mean, this boy was unfortunately on palliative care, and so the clinician felt that basically they ran out of options, and so they tried it. Well, it turns out it had a very rapid response, very broadly. The clinician and the physical therapist really saw very notable, very meaningful changes. And the boy's alive today, about 15 months later. Which he was not expected to live that long. And he continues to improve. And now we're actually looking at kind of second line medication for him that the clinician could explore. On the right is actually KMT2B dystonia, so very different disease. There's dystonia, which kind of muscle kind of spasms and muscle tension problems, but also very severe neurodevelopmental disorder, cognitive function. And this boy was nonverbal for 4 years, had a lot of sleep problems, ADHD, and other things. And we found it's a prescription drug, but we just anonymized it here as Ravel 68. But the clinician prescribed it, and basically so many of these factors have improved within weeks, I mean, within really days of treatment, and actually culminating in 4 months later, him actually starting to use sign language to communicate back with his family, which was very, very impactful. And so now where we stand today, we have over 45 programs. Again, we keep adding new programs all the time. Several of them are in this clinical proof of concept stage. We're actually about to start running 2 clinical trials in Rett syndrome and Pitt-Hopkins. And Pitt-Hopkins, actually, the foundation is sponsoring their trial. Again, we just passed the costs along. That's for vorinostat and like a special formulation of it that we developed. That's transitioning more towards the commercial side, but then there's a lot of off-label use. But then for most foundations, we do a lot of research, the squishy-ware tadpole models, which I didn't go over, and certainly mouse validation with Jackson Labs and other groups. And so in conclusion, what we have here is really this rapid therapeutics prototyping approach. We can ask the question, does it work, right, in an actual patient using existing molecules? We generate the data, define the patient state, predict therapeutic mechanisms, interventions, and then validate. So both in animal models, we heavily collaborate with other researchers in academia and in industry. And then one thing that we do or can collaborate on are these very low-cost clinical trials. What we can do are formal clinical trials. We can do any sort of research, but we're also happy to collaborate with clinicians through observational studies or just clinical care. The idea is we want to de-risk the therapies, right? We want drugs that actually make a meaningful difference in patients' lives. And so what the impact of RareShift is on the PDE community, one, you'll have primary RNA sequencing data, right? So this is not from models, this is not from derived lines, which all of those, by the way, can be very useful, but they exclude things like in any in vitro model, the impact of the whole body, right? Diet, sleep, all those things are really not captured in any sort of cell model. And even the organ and tissue level interactions are typically not. But so this is directly from patients, basically 20 or more patients and their matched controls, and all this data is shared. We share everything for research use. There can be potential treatments identified, so again, repurposable candidates, as well as potentially some of the research use or kind of high-risk prescription medications that we'd have to do additional work. Those would not be easy to translate into the clinic, but could be very effective, right? And maybe a new drug is the right path there. And then the bottom line here is this third point, which is so critical. You need to understand the patient population, and especially there's all these really exciting research trajectories by various groups and all these other diseases. The reality is that most patients with a given diagnosis may not respond to any one particular treatment. And wouldn't it be great to know that now instead of, as the outcome of a clinical trial in 5 or 10 years? And so that's really what we're doing, discovering treatment paths, finding ways to mitigate any potential adverse effects, including, by the way, in gene therapy. We can see all the cocktails of drugs that patients are given. We can see patients responding positively or not. And then just optimizing that strategy for the path to the clinic. Is PDE one patient group, which it could be, we have seen that. Is it multiple, 2, 3, 4 subgroups? It could be. Well, again, we have seen that. We don't know until we do the analysis. And so that's it. That's RareShift in a nutshell. And again, it's about function first, right? It's function first drug development, starting with patients. So happy to answer any questions that you might have. I'm here.
Thanks, Richard. Hey guys, sorry I was late. Hey Christy, thanks for hosting. I was able to listen to the majority of it. Thanks so much, Richard, for presenting. Christy, you want to catch us up on where we started?
Hi, Anna. So we were just talking a little bit about, we didn't go into the depth of like what CurePDD was planning. So he presented his, what the company does, and I was hoping you would show up and could talk for CurePDE because I didn't want to do that.
Yeah. So we decided to proceed, and I don't know, Richard, if you mentioned that there's the 2 models of doing the analysis. There's individual model of analysis where you compare basically one patient to their one healthy control. And then the other model, which is a group model, which is that you can take up to 20 patients and up to 20 healthy controls, which is what we
Minimum 20, yeah.
What?
Yeah, minimum of 20. Yeah, we
Oh,
started.
minimum of 20. Oh, okay. I thought it was maximum of 20, even better. So Cure PDE decided to go for the group analysis just because we thought we still really don't know if there's subsets of patients, and we wouldn't even know how to pick just one person, and that isn't really what we're after. And so what we want to accomplish and what we've started reaching out to a few folks about, and what we really want everyone who's interested to participate in, is helping us sign the consent forms, and there's informational videos. I think I can send out that email with all of the links. So there's videos for all of you guys to watch on how the consent works and how the swabs work. And I believe they can be mailed anywhere. So anyone who's in the EU, I believe that's not a problem. And just reach out to us, and we'll post information on our bigger Facebook page and on the other PDE channels as well, because we want this to be available to anyone. They do have to have a confirmed genetic diagnosis that needs to be shared, right, Richard? That's one of the few kind of like.
If it's not confirmed, that's fine. We just need to know that it's part of this group somehow, right?
Okay.
So
Okay.
that's okay.
Okay.
We do not use that information initially at all. We just the RNA. So we work with undiagnosed patients as well.
Oh, okay. I
Or
gotcha.
these complex, like multiple mutations, and then which one are you? It's your unique case at that point.
Okay. Okay. So computationally, you're fine with that, with folks not
Yep.
knowing there, because I know we have some people who haven't been officially maybe sequenced, who are a little bit older and things like that. So
That's fine.
yeah.
We do ask for medication supplements for both the patient and the control, because we will absolutely see that in the data. And so if something comes up that a patient's in a specific subgroup or something, we always look at would the medications or other, again, just supplements are included in that list. Would that help explain their case? Sometimes it does, sometimes it doesn't, but
Yeah.
we absolutely have seen certain cases where the medication is very much explaining what's going on. And so we need to just correct for that.
Cool. I don't want to take up all our time. I want to leave questions if people have questions. I know we have some smart folks on the call, so anyone is welcome to ask a question, whether it's about the basics of Unravel, or a question of me of how we're thinking of doing this, or really anything. So we can have as many conversations as you guys want on this topic. This doesn't have to be the only one, but this would be a place to have Richard available to answer questions as well. So anyone is welcome.
Hi, I'm Christina. I do have one question, and I'm sorry, I may have missed this. I was having to entertain my 2-year-old in between listening. But whenever you're predicting this data, are you going in and looking at picking a certain drug class, for example, and then getting the data based off of that drug class?
No. So this is just based entirely on basically RNA signatures of the drugs. And so what I mean by that is that there's some cell lines typically that were dosed either by us or another group with that drug for some amount of time at a certain concentration, and then sequenced, right? So it's telling us how are the genes changing in response to, my go-to example is aspirin, because everybody knows what it is. Almost never comes up. And so, for example, what is aspirin doing? Well, okay, we know it's a COX inhibitor. That's like how it's classified scientifically. That's great. It has a whole bunch of other activities. It's a pretty dirty drug. That is what we use. The fact that it hits all these other pathways, all these other things in a very specific way versus, let's say, ibuprofen or Tylenol. Those are all very same class of drugs, but very different in terms of how they act. We take that underlying RNA information, we ignore what those drugs actually are known to do for the calculations. Now, later, when we are looking at what are the groups of drugs, we want to see, are there any known mechanisms that, for example, COX inhibition for aspirin, are all the same drugs in that class coming up as hits, in which case it's probably COX inhibition. It would actually be very helpful to know that. But sometimes that's the case, sometimes that's not the case. And so But for the initial discovery, no, we don't make no assumptions about what a drug is doing or not doing at all.
Um, how do you narrow it down then, or do you have to narrow it down?
Yeah, you do have to narrow it down to be able to advance it. Even into animals, you can't screen thousands of drugs. And so what we've been able to do is we narrow it down typically to kind of 5 or 10 in the end. Like not the result of the study will be, the full drug list for usually a couple hundred drugs ranked from most therapeutic to actually anti-therapeutic. That's where you get the toxicities and like the ones that are making the disease worse somehow. Out of those, we tend to look at, okay, can we get to like 5 or 10 drugs? The reason for that is that the worst we've done on translating a drug into an animal is 1 in 2 success rate. That's the worst we've done. Which means if you have 5 drugs or so, you're going to get some hits.
Okay, have a good one.
I just want
If
to ask if you could compare to other things we talk about. So we have screened every FDA approved drug
Great,
in our animal models for PDE. So we
great.
have done all of them, and we have 13 hits. So I wonder if you could compare and contrast sort of how
Oh,
those screenings
yep.
happen in animal models compared, which we are using a different endpoint than you are for the families.
Yeah. No, we love doing that. So this is what we really always ask for. What other data might exist that we'll give you that our drug list regardless of that, right? So you can blind us until then or not. It's totally up to you. But then seeing how does that prediction compare with anything else that maybe generated. We love doing that. So we've had some really good correlations with fly studies. Again, they tested a whole bunch of molecules like with the Prestwick library. If you're familiar with that, just again, a lot of just repurposable drug candidates. And there's a good amount of agreement. Now, it's not perfect because also in the animal models, you're not going to get the medication, you're not going to get all the other stuff. They are animals, they're not the human, right? There's going to be some subtle, maybe not so subtle differences. But you should expect some overlap, and we do see that. And those tend to be the ones that you want to prioritize because, hey, now you're kind of hitting all these different reasons for why a given molecule should be advanced.
I don't want to interrupt Professor Gosby at all. I just want to remind the families that we're looking for something different, right? Like our endpoint is death because those animal models die. And so we're looking for drugs that rescue the animals from dying, which could be a completely, like, could not be the thing that your kids need. So it's like it could be a very different approach.
Mhm.
I know we've had these meetings before where we said, hey, we've screened thousands and thousands of drugs, and we have because it's an easy to do in our model system, but it also might be different. And I'm going to defer, as always, to Professor Gosby.
Please, Curtis, it's not necessary. So I'm just curious about sort of the dirty part of this, as far as the type of data that you're getting from nasal swabs. Let's say that you have a patient and their sex match control, a little girl and her mother, for instance, and one of them is, suffering from really serious nasal allergies at the time her swab is being taken, whereas the mother is not. And so there's there's going to be a signal from this completely unrelated process going on in her nose that mother is not experiencing. How do you deal with something like that?
Yeah, no. So in general, we recommend people, for example, to not have any active infections. Like we had a couple cases of COVID where, COVID happened in between them deciding to start a study and then actually receiving the kits for swabbing. So we held off up to that point. There's the assumption that it's not related, which sometimes is true, sometimes is not. We work with a couple cases where there are these autoimmune disorders, and turns out they actually may be related to some of the underlying genetics. But it might be a kind of a unique case within that disease. It's not necessarily common within that disease, but could be related. But you're absolutely right. I mean, so this is where when we look at the population level, that's why we ask for 20 patients or more because there's just more diversity, more heterogeneity, more noise, partly because of the genetics, partly because of the environment. And I would say allergies fall into that environment, but also their genetics and other aspects are making them more susceptible to it. So this is what we do all the time. Now, what's interesting is, if I should go back a couple slides to this one. So one thing I want to point out is actually this, we did UB 144 is a patient control pair. And so it's about a 10-year-old and a 6-year-old brother. But they were actually sampled twice, about a week apart, and they fall in the same group. So there's a personalized study, which is, we sample over 2 days. It's 8 times as much data per person. And this is just a single time point that we collect for the population studies. We actually get very similar results out. And so the rationale here is that because we're looking at these networks of genes, not just genes going up or down, but these networks, we can make inferences that are more abstract. And in your example that, if let's say this patient had allergies, it would absolutely skew some of the results in terms of you probably would get more antihistamines and other drugs popping up. But a lot of the other mechanisms would not. And so those would be very consistent. And then we're looking at it in the context of a patient population. These are not identical drugs. The only identical drugs are basically in this identity line right here, where it's the same list of drugs in both cases. We tend to see that there are some drugs that are highly ranked for all these patients within this subgroup, and then we would focus on those. So in your hypothetical example, where let's say one of these patients had seasonal allergies, yeah, they might have high antihistamine ranks, but maybe those don't show up for any other patient. That's how we would exclude it. So you're absolutely right. I mean, again, that's the issue of dealing with any clinical scenario. That, like in vitro, you can eliminate a lot of those factors, but then you're missing a lot of other things. In terms of just general infections, this is also why we tend to, again, if what we recommend, and now, is it always available? No. But the control is in the same household as the patient, because you're exposed to the same sort of pathogens. Now, you might have very different responses as a result. That could be because of the underlying mutations, right? And maybe not the pathogenic one, but potentially some other ones. And so, but at the same time, you're still exposed to the same viruses. Like I have 2 kids, and they're little bioterror factories, and they bring back stuff. But my wife and I, for example, have different outcomes, but we're at least exposed to the same thing, and that we have seen that anecdotally be fairly well controlled between household relatives.
So as far as recruiting subjects for this, I heard Anna say that there would be promotion on the foundation's website. I'm a retired child neurologist. I still have contacts with several PDE families, I think many of whom are not part of this group. I could certainly reach out to them and inform them about this opportunity, but I would need to know where they would then go to get consented, and who actually does the consenting, and who is responsible for the study.
Yeah, so this is what we set up for RareShift. So we have a kind of an overarching IRB for this whole study, and sort of independent of diagnosis, right? So independent of the genes, because our whole premise is that a lot of these therapeutic mechanisms span diagnoses. And that's actually how we're making rare disease commercially viable, is that by unifying different rare and ultra rare diseases together. And so we have this IRB. We do have the consent forms for both participants, so both the patient and the matched control. Typically, it's a parent. That's the most common one. Same sex is quite important. In some cases, that's not possible. Do the best you can. We're happy to respond to any questions. And we also have a registration form where that's where all the drug information, diagnostic information, and shipping information is contained. So that's sort of the personally identifiable piece of it. So we handle all of that, all the consent, clinical study aspect of it. The recruitment, we work with foundations, families, whoever it is. Essentially, the data inform the questions being asked. The model does not. We don't model disease. We don't artificially ask specific questions. And so the recruitment process is really critical in that regard. Here, Anna, again, don't want to put words in your mouth, but I believe you agree that the goal is to really do a population assessment. Let's see what the whole population looks like, as opposed to, in some cases, we want to look at very specific mutation subtypes, right? Again, different foundations have different goals. And so here it's more of a kind of all comers. So anybody with this gene that's somehow impacted or has a suspected diagnosis, I think sort of would qualify. And let's see how it shakes out. That's essentially the goal of this study.
And said the swabs get sent to folks' homes, so it's an at-home collection kit.
Or wherever you tell us. There's an address, as long as you can get FedEx or other
Yeah,
shipments.
but you're not going in anywhere to get any sample. It's a self-sampling situation. So.
Yeah, and there's a video, like instructional video. The instructions are a little bit different than from a COVID test kit. That's why we made that video, but it's a soft nasal swab. It's not too crazy.
I guess one of the other concerns I have is that many of these patients and their parents, while they have been genetically confirmed, may not really have that information available to them. And so, how would that, clinical data be released to you?
So we do have, I mean, if they have any sort of genetic test results, they can either input it into a form, or we also have an upload option if there's just a document and just share it. We can.
That's my point. I think many of these families don't know their genotype. They know they've been confirmed, but they don't have access to that raw information, which is very technical and potentially not of very much use to them, other than knowing that the diagnosis has been confirmed.
Right. So we can't help on the access to that information, but patients have, in theory, should have a right to that information. They could pass that on to us through that form.
You don't need it, so you don't need the specific mutations if they
We don't need it. We just need to know that a given
But they're
patient is
part of our
group.
group. Yeah, yeah. I think Richard had said that they don't, if it's not available, for example, I was chatting with somebody and they don't have their report, and it's, I guess, as long as they are relatively confident that they have PDE, then otherwise, I guess their signal would be completely off anyway, and they probably wouldn't.
We'll see it. Yeah, we'll see if they're totally off. We've dealt with this a couple times. There's one group where there was actually a related gene that sort of, there's a family that doesn't have a foundation. They're just piggybacking off of a related gene. So they participated and actually were quite similar. And there's one that was undiagnosed. They're like, symptomatically we think it's this, but they were in a very rural, remote part of, I forget where, somewhere in Asia, I think, and then they just never had a diagnosis. But we could see how that aligned or not.
So if folks want to participate and they're comfortable with the consent, I think they can, in theory, provide as much or as little detail, right? I mean, obviously more, and Jen was mentioning, like, if kids are on vitamins, then that's important to know, is like all of the stuff that folks are on, because it's going to change the way that their profile shows up. So for the
It'll
kids,
help explain, it won't change the results, but it'll help explain the results.
right,
Yeah.
right.
So and again, going back to the diagnostic data, so we do share the anonymized data back, right? So no personal information. They're just like these UB IDs. You don't know who it is. But the more data that's there can be very helpful. So if you do have whatever your diagnostic information, even the technical pieces, we just add that to the database so that it's there for research use, which it could be quite useful, right? But if you don't have it, it just stays blank. I mean, that you can still participate and see where you are in that study without that information, but it is helpful, and just from a scientific standpoint, the more the better. The drug supplement side, very, very helpful, because it can help. We have seen patterns there quite easily when we have that information.
And I think it's interesting because in PDE, I think we do have categories of folks, especially some of the younger kids are in a very similar kind of category of treatments. Some of the middle folks are on, something a little bit more reminiscent of simply B6. And, then there's a category of folks that maybe didn't even get early intervention that are older patients. And I assume that'll also come through. I guess for me, and for Anastasia, what we're excited about is we're not trying to oversell this in terms of, and I think Richard did a good job of not overhyping that there's some sort of miracle drug coming at the end of this, or, you know, that we're going to find something that's, you know, that there's not some pot at the end of the rainbow, but I do think it has the potential to help us understand not only the categories of kind of PDE groupings that we have. Are we all generally similar? Are we slightly different? Are we different along, you know, something that's easy to understand? You know, are we all generally responding in a similar fashion? Are we all generally different than our controls? And one of the things I like that Richard had told me earlier was that this is not so much mapping like healthy to diseased, but really understanding. I don't know if you remember how you explained it to me before, but really understanding like the roadmaps that exist within the different groups, right? So, and how can we use those roads that we have inside of our PDE kids and adults, and kind of understand if there are available treatments that can enhance their health. And yeah, so it's not necessarily to get them. I don't know. I guess it's just a different mind shift for me of like going from disease to healthy versus just, yeah, anyway.
Yeah, because fundamentally, you know, every patient, you kind of gave that Google Maps analogy, which is every patient is in a slightly different starting point, and the destination may be a little bit different too, because again, it's their sort of matched controls that we focus on. It's not exclusively that, but that's a good way to think about it. It's sort of how do we make the patient like the matched control at a high level. Each patient's starting in a slightly different spot, right? So if you think of like Google Maps, and again, I don't know where everybody is, but I'm here in Boston. I live in the suburbs and trying to get downtown. I use Google Maps every day because of traffic, right? It's not because I don't know where I'm going. My kids make fun of me, like, wait, do you not know how to get to work after all these years? It's like, no, it's because the traffic changes, there might be an accident. And then that can have a very different impact for, you know, a colleague of mine that lives one town over, and there's a real example. She might get to work, you know, 20 minutes before me because the accident happened like just between us, right? And so that's what we see with these patients, is that everybody's in a unique spot, and we try to understand diseases from the collection of individual patients. Now, sometimes they're all the same in terms of the response, and that's actually a great scenario from a foundation standpoint, because one therapeutic mechanism or several therapeutic mechanisms but could all work in that population. The reality is that sometimes that's not the case. And again, we don't know going into a disease if that's going to be the case or not. With SYNGAP, for example, I gave that example. There are some pretty big subgroups by severity, by seizure, you know, likelihood and severity and other aspects. But one thing that we saw there, all the mechanisms that we predicted were actually being explored at the conference in December. What we also see is that 100% of patients will not respond to any one of those mechanisms. So now, as a foundation, if you're thinking about translating all the hard work that you've put into it, all the funding that went to advancing that research, what does that clinical trial look like? Who would you bring into that trial, right? And there are going to be patients who will absolutely respond to some of those mechanisms. The data and the science are great. The reality is that maybe came from a mouse model, and maybe that mouse model represents, I have no idea for PD, we don't know yet, right? But we also work on the mouse model side. How representative of the mouse model of the population? It may be that it's extremely representative of half the patients, right? Great. Wouldn't be like, if you know that, you can absolutely work with that and advance the programs. If you don't know that, you're in for some sometimes unpleasant surprises. And so that's kind of how we can use this kind of predictive information. Again, it's not about like, here's a magical cure. Like, well, how do we advance all these efforts in a very efficient way to the clinic? Because ultimately it is about the clinic. We do have to go into patients at some point, because that is the problem we're trying to solve, not how do we treat a mouse, which again, that's a critical intermediate step. But ultimately, I think we're all aligned that we have to help the patients, and that's where we're aiming, right? And so how quickly can we get there? How more efficiently can we use resources? I think it's in everyone's interest.
Anybody have any more like concerns that they feel about it, or something that makes them uncomfortable, or any sort of hesitations that we should address as a group or be aware of, or even if you've heard, something that might kind of detract folks from wanting to participate, or I don't know, just any general feedback.
I just have a question. Both my kids still have runny noses from being sick. Should I wait till they don't have runny noses to swab them?
Yeah, it depends on your kids. Like my kids last year went, I think, 0 days without runny noses. So I think just you have to kind of judge how severe it is. I mean, if there's any fever or anything like active infection, I would absolutely not swab.
Okay.
If it's your
They
kind of
had the
typical,
flu last week, so there's
yeah,
no
I'd
fever
hold
this
off,
week, but
give it another week.
okay. Okay, sounds good.
Yeah, but just kind of your
Awesome.
sniffles, your kind of, general little kid runny noses, that's how it is. You're never gonna, you have to wait probably a few years for that to go away.
Okay,
So,
sounds good. I'll give it one more week and then I'll do it.
yeah, I mean, you decide. I mean, if it's, if they're sick versus like just kids.
Okay, sounds good. Thank you.
Any other questions, guys, since we have Richard here? If not, I will post the full instructions, and I can send them to Sid and Curtis and Philip by email, in case you want to pass those on. I assume, Richard, if anybody has questions, if any of our science folks want to get in touch with you about some, other ways to correlate data or give any background data or anything like that, they're free to email you.
Absolutely. Yep.
And yeah, we'll move from there. And I assume we just check in with you to see how many folks have signed up to now, or is there a way to kind of keep us updated on our
Yeah,
number?
so we just started that. So actually, we're literally just doing the first round of project updates for you guys. We do
Okay.
every 2 weeks. Yeah, just a note for everyone. So everything's confidential once you basically enroll in the study. So we do not even share the names of participants, like the foundation or nobody has access to that besides us that are part of the IRB approved team. And so all the communication kind of has to go through us. Like if Anna asks, like, okay, who all has signed up? I can't and won't share that.
So
And
you can
so
give
it's
me
sort of
numbers, right? You can give me
by the
projects.
numbers, exactly. We have 5, we have 6. It's 6 actually, by the way,
Yay,
as of
six.
yesterday. Because I had a feeling I would get asked that question. So we have 6 who signed up. Again, we don't have the samples back or anything, but just who've signed up and are participating. You're also welcome to withdraw from the study at any point. I mean, once you're already in the database, the consent doesn't let you really withdraw because everything's done. But you're anonymized at that point anyway. But anything up to that point, confidential. The email that I sent Anna has my email, and then Bridget, who's the clinical research coordinator who does most of the kind of day-to-day work on this study. She's there. Anything related to us, if you email us, that's all strictly confidential. So any questions, anything like that. I know sometimes we get some questions around controls. There's some sensitive family situations. Happy to answer any of those questions privately. We've probably seen every scenario at this point out of the 1,500 plus people. So just let us know. We're here. Yeah, anything like Christy mentioned, questions around the like runny noses, any of that. If it's outside the U.S., customs has been the last few weeks, especially, is going to be a little bit changing. So we've run into some issues just last week. But so let us know for any international ones. U.S., there's never been any issues. But yeah, any of that, just don't hesitate to
New
reach out.
tariffs launched on RNA samples across the border. I hope there's no new tariffs launched on RNA samples across the border.
You'd be surprised. It's already actually impacted us, actually, like just yesterday, and then they reneged on it was Canada, shipping to Canada. Geez, like as mellow as it gets internationally.
You got in before the ban.
And yeah, when we actually got asked to fill out all this extra paperwork and everything for sending a couple swaps to Canada. So
I will just suggest that if you do get some European subjects who are also enrolled in a similar study where we're doing both blood RNA-seq and making organoids out of their fibroblast line, if they ask the difference, we used non-related controls. And I think we had 50 responded individuals so far out of the US.
Hmm.
So hopefully you can get, I mean, it seems like there was high uptake.
Cool, great.
It's great. I mean, but we routinely work all around the world. I mean, that's not a
Yeah.
problem. And by the way, these samples are not considered clinical samples because it actually inactivates any pathogens. They're not considered infectious, hazardous. There's even like a CDC certificate we don't routinely include because most customs groups don't need it. But we haven't had any issues with. There are a couple countries that we cannot ship to or from, depending on a couple circumstances, but it's very few. But US is easier, always easier, but we're happy to work internationally.
Okay, guys, so just keep a lookout on the Facebook group then, and maybe through other folks, if Curtis and the team volunteer to send out emails. I don't know if there's any sort of conflicts there, but don't expect there to be. So, okie dokie.
And if you're enrolled in our large natural history study and you don't have a copy of your genetic results, we're happy to send them back to you. Just email Christy. But anyone who's enrolled in the natural history site through our site, we're happy to give you back your genetic result if you don't have it.
How do you enroll?
In the natural history study,
Well,
also email Christy.
For this, Scott, I'll post on Facebook and I'll send you a message. You just have to sign an online consent form. If you want somebody to walk you through the consent form, let me know. I'd be happy to. And otherwise, they just sent you something to your house and then you swab your nose and send it back, and that's how you enroll.
So what is this sample being used for? I mean, what is there a particular drug or something?
That's what we're trying to identify if there's so we're actually looking for potential drugs.
Yeah, so the sample is your cells. If it was, let's say, you participating either as a control or as a patient, it's your cells inside your nose, and then we specifically look at the RNA, so the RNA molecules. And then we can use that through our platform to figure out, are there any drugs that would be helpful? But it's not a drug test directly. But yeah, that's kind of how it works.
Well, certainly you must have something in mind. I mean, you're not just collecting all this together. You're thinking about looking at something, aren't you?
So what's really exciting is that we don't have to have anything in mind. Historically, we've been really bad at picking something that's successful in the clinic based on assumptions. And so we actually simulate 40,000 different molecules on every patient. So we just test them all in a computer. We don't assume anything about any one mechanism or patient. And that's actually what makes this really attractive, that we can find completely new mechanisms that don't necessarily make sense initially.
So if you've done this before, how successful do you think that this could be?
So far, we've been quite successful. So in individual patients who have taken a drug with their clinician or off-label or with as a supplement, 6 have reported back and have been actually added long enough to see any effects. 6 out of 6 have had positive responses. We've predicted every group's research programs, basically 100% of them. So we published some work with COVID patients, and there we had an 80% predictive success rate in like 4,000 patients for 5 different drugs. So it's quite effective. Certainly better than the kind of up to 5% success rate for typical pharma development.
All right, guys, I need to jump. Dang job needs me at 1. Richard, thank you so much. Does that mean that I am free inside our Facebook group to post both of the email addresses on that email that folks can get in touch with you?
Absolutely. Yep.
Awesome. And thank you everyone for joining us, and we can keep having conversations as needed. So, I don't know.
Thanks. Great to meet everybody.
Bye.